Okay. So a reminder where we are. So we've walked through the layers of the general walls of the GI tract, talked about some of the cells involved. Then we talked through the order of kind of regions of the GI tract running from the mouth all the way down to the anus. We talked about associated glands that we're going to see as we start to think about the chemical digestion of macromolecules. And last time we began thinking about that digestion process a little bit more in detail, starting with our carbohydrates. So we're in our absorption packet looking at carbohydrate digestion, thinking about how this begins in the mouth with the digestion often of starch, which is a special way that plants have of storing glucose. So we had broken up our starch partially. So when we have our two types of amylase act, we can cleave some of these bonds, but what we're left with are a bunch of these corners, basically, where we have these branching points. We're also left with disaccharides just because of the way the amylase works. We can't quite get to each individual glucose monomer. So before we move into our proteins, I want to do a little bit of a thinking question here about some of our carbohydrates. So we mentioned the existence of the brush border in the small intestine before. So another enzyme we have related to digestion of carbohydrates is lactase, which happens to be one of those brush border enzymes. So lactase in particular breaks down lactose, which is a disaccharide. It works the way that these other brush border enzymes work. So where would we be looking if we were trying to find that lactase, that enzyme? Take a quick look. Okay, I got a couple of guesses. Okay, so lactase being a brush border enzyme. Remember when we were thinking about the surface area that we're creating in the small intestine? We mentioned that we have villi, these projections where we'll have capillary beds as well as lacteals. And then if we zoom in on an individual cell here, we see the microvilli on this apical membrane. And then on the blood side, right, where we'd have these capillary beds, that's our basolateral membrane. Okay. So when we're thinking about the further digestion of lactose or any other disaccharide, like we were left when we were digesting salt with maltose, another disaccharide, what we're going to need to do is as we're passing those disaccharides past these cells through the lumen of the small intestine, they're going to need to come into contact with this brush border made up of those microvilli. So this is where we would find lactase. So if you are lactose intolerant, this is what isn't working. You don't have this lactase enzyme in that brush border. All right. So what it should be doing is breaking down lactose or disaccharide so that we can then pull those monomers into the cell, and then they cross that basolateral membrane into your bloodstream so that you have fully processed this carbohydrate and can now start to break it down for energy, incorporate it into other cells. Okay. So that's just one example of a way that we would continue to break down carbohydrates after we pass that initial bit of chemical digestion, beginning in the mouth and continuing as we enter the duodenum and start to break things down with pancreatic juices, including pancreatic amylase. We do need to do a little bit more with these brush border enzymes, because when we're thinking about carbohydrate absorption, we're only going to be absorbing monomers, so individual subunits. Cool. So next, we're going to think about the chemical digestion of proteins, which is going to work a little different. Okay. So when we think about proteins, we are going to be thinking about a lot of different enzymes that have to work together to break down these really large molecules. So we're going to have many different enzymes that work in many different ways. We have many different amino acids, but we can generally split them into a couple of classes. So our general word for an enzyme that breaks down a protein is going to be a protease, right? So we've got that protein part that tells us we're breaking down proteins. That ACE part tells us this is an enzyme. Okay, so proteases are enzymes that break down proteins. Okay. So in general, I got a couple things going on. So I'm going to actually talk about the zymogens first, because we mentioned this when we mentioned pepsin in the stomach. So our zymogens are going to be like an inactive form of all of our different types of proteases. Okay. They're stored in places called zymogen granules. So we've got a storage location for them. And when we're ready to use those zymogens to start to activate them, they're going to get secreted from a cell through exocytosis. So that process we've seen before, where we have like a vesicle, but in this case, we're specifically calling it a zymogen granule, coming towards the membrane, bursting, letting out, in this case, those zymogens. Okay. And then they're going to need to be activated by proteolysis. Right? So zymogens are enzymes. They are themselves proteins. So we see that proteo for protein, lysis for breaking down. So to activate a zymogen, we have to break it down a little bit. So when we think about that breaking down process, whether it's of a zymogen or a larger protein that you might have eaten and you're trying to absorb, we might see these other two groups, the endopeptidases or the exopeptidases. Okay. So when we think about the protein, we're going to unfold it largely by denaturing it using our stomach acid. So we're going to be left with a polypeptide, right? So we've got our little bead-like amino acids all in a chain, right? So our endopeptidases, they're going to be able to break kind of the middle bonds, right? So endo is like inside, peptidase. It's telling us again, this pep is for our polypeptide, ACE is for our enzyme. So we're going to be able to break like here if we're thinking about an endopeptidase. So they're able to split our bigger polypeptide into smaller little chunks by attacking those interior bonds. Our exopeptidases, exo is like for outside. So what our exopeptidases do is they help us peel off the amino acids on the ends, right? So we need a different type of enzyme to do this part once we have everything in small chunks. So those are generally how we might group together some of our different types of proteases that we're going to encounter as we move through the process of protein digestion. Okay. So applying this to a specific enzyme. So one of our proteases that we're going to see is an enzyme called trypsin. So if I tell you that trypsin is activating other proteases, what type of protease is it that it would be activating? So how would we class those enzymes that trypsin acts on? Yeah, so it's going to be the zymogens. So the inactive proteases that we're going to need to activate are all together just going to be called zymogens until we activate them into specific types of proteases with specific actions. Okay. So if we're thinking about protein digestion, walking ourselves through the tube, right? So say you ate a burger, we're trying to break down that burger patty. You chewed it, so you did some mechanical processing. The chemical digestion of the protein in that food isn't going to really substantially start until you hit the stomach. So unlike our carbohydrates, which we start digesting even chemically in the mouth with those amylase enzymes, we're not going to start proteins until we hit the stomach and hit pepsin and our stomach acid. Okay. So we talked a bit about the gastric pits when we looked at the structure of the stomach and some of its specializations. So remember, we have these chief cells and parietal cells in the stomach. Okay, so pepsin is going to be really our first enzyme that's going to help us start to break down proteins. It's going to be located in the stomach, and it's coming from the pepsinogen that we secreted from these chief cells in the gastric pits. So our pepsinogen would be coming out here, right? Gets into the lumen of the stomach, so the hollow part. And then we're going to need to activate that pepsinogen into pepsin, right? So pepsinogen would be like one of our zymogens. And once it's activated, we've got this enzyme pepsin. Okay. So that activation part is going to come from the actual chemical environment of the stomach. So that acidity of the stomach that denatures proteins, so starts to unfold them and break them down, that in and of itself is enough to start to break down that pepsinogen, which pops it into its active form. Super convenient for us. So a reminder, that acid, that hydrochloric acid, is coming from the parietal cells, so that other type of cell here that we were focusing in on in those gastric pits are secreting hydrochloric acid. So those hydrogen ions are what creates a low pH, creates that high acidity. So that's also going out the gastric pits, ending up in the lumen of the stomach, and having the end result of activating that pepsinogen into pepsin. Okay. So pepsin is then going to start working on the proteins that you've eaten, and it's going to start that breakdown once it's in this activated form. So here we can see it acting like an endopeptidase, here breaking up some of those interior bonds, right? Breaking up a larger protein into these smaller little peptide fragments. So we're going to end up with these peptide fragments mixed up into our chyme. So a reminder that the chyme in the stomach, that's your food that you've chewed up and started to process, mixed in with the gastric juices that the stomach has secreted. And that's what we're going to be slowly letting move into the duodenum through our pyloric sphincter, this kind of control muscle that we have at the end of the stomach. Okay. So then we're going to hit our small intestine. So first the duodenum, and then we're going to move into the jejunum and the ileum. Okay. So our protein digestion is going to continue here. We're not done. So far, pepsin has just broken up our polypeptide chain into some smaller peptide fragments, but they're not, for the most part, small enough to actually be absorbed across the wall of the GI tract. Okay. So here's where we're going to have a lot more enzymes come into play. So what we're looking at here is the lumen of the small intestine. So your GI tract is coming down this way. And we're zoomed in on that part of the duodenum where we have our pancreatic duct coming in. So a reminder, our pancreatic duct comes in, attaches or has its opening into the duodenum. So as it is entering here, not pictured, we'd have it meeting up with our bile duct, creating this ampulla of Vader. And ampulla is just like a swelling. moving through the sphincter of Oddi, which would be located right about here at the entrance, and then entering the lumen of the small intestine from the side. So this is our accessory glands starting to pour additional material into our small intestine. Okay. So from the perspective of protein digestion, this is going to be really important because there are a bunch of pancreatic proteases. So the pancreas has made a lot of enzymes, many of which are for breaking down proteins, although we know that we also have our pancreatic amylase we've discussed for breaking down carbohydrates. So the pancreas is really going to help us break down pretty much all our different types of macromolecules. Okay, so coming down, we've got our chyme and those peptide fragments coming down the tube from the stomach. And now we have our pancreatic proteases coming out of our pancreatic duct. So we're highlighting a couple of important ones here. So the pancreatic proteases that we're ultimately going to be interested in are listing them right now by their active forms, right? But you can see that the pancreas has emitted this trypsinogen, which turns into the active enzyme trypsin. We have our zymogen chymotrypsinogen, which turns into chymotrypsin. So that's the next one on the list here. And then we have procarboxypeptidase, not following that same kind of word ending, but we're also going to have to activate that one. So that one becomes our carboxypeptidase here. So those are named by their active forms here. So we can see a little bit about how they got activated as they were entering the small intestine. Okay. So coming out the pancreatic duct, we're going to focus on that trypsinogen first. Right, so our zymogen trypsinogen here got activated as it ran into this brush border protease enterokinase. So this is another brush border enzyme. So you can see it kind of embedded, or at least sitting on top of the wall of the small intestine here. So when trypsinogen runs into this enzyme breaks it down a little bit, converts it into its active form trypsin. Okay. And then as we saw in that previous Slido question, that trypsin, one of its big jobs is to go and activate the other proteases that we have coming from the pancreas. So you can see it do it. All right, that's how we got our chymotrypsin. That's how we get our carboxypeptidase. Okay. So this is the general flow that we have happening in order to activate these pancreatic proteases. Right, so our pepsin got lucky in the stomach, it can just get activated by that hydrochloric acid in the stomach, but we need actual enzymes in order to activate these pancreatic proteases. And once they're there, they can then act as endopeptidases and exopeptidases, depending on which ones we're talking about, in order to continue breaking down our polypeptides and those smaller little peptide fragments into pieces that are small enough for us to absorb across the lumen of the small intestine. So aminopeptidase is not pictured here, but it's another brush border protease, kind of like the enterokinase, that's going to actually help us continue that digestion of peptide fragments. So that one's not usually activating proteases, works a little different. Okay. So we're breaking down the polypeptide chain using our endopeptidases into these smaller fragments, these smaller peptide little bits. We can use exopeptidases to attack those peptide fragments from the outside, make them even smaller. Now we're going to absorb those amino acids across the wall. Okay. So we have a couple ways we can do that because we know we have many different types of amino acid. But in general, one of the things that we're going to be doing is we are going to be using our sodium-potassium pump to create a sodium gradient, right? So we can see that happening here. That's a picture of our sodium-potassium pump. So in this case, we'd be thinking about the extracellular fluid here, really facing the lumen of the small intestine. right? If we were to specialize this to think about absorption in the small intestine. So this would be the inside of the intestinal cell. So we're going to be creating, oops. Yeah, we're going to be creating a sodium gradient in order to pull stuff across. Actually, maybe don't think about the lumen being on this specific sodium potassium pump. Sorry, that's not as convenient. Anyway, we're creating a sodium gradient is the point. So then once we've created a sodium gradient, we can use that sodium to pull things across the wall of a cell. In this picture at the bottom, we saw before that we could do this with glucose in cells in general. We can do secondary active transport in the same way on a bunch of our amino acids as well. Once we pull our amino acids into the cell, we can then allow them to filter through the basolateral membrane, often through facilitated diffusion. So in general, if we're thinking about this process, because of the number of different amino acids we have, there would be many different transporters in cell membranes that are adapted and designed for pulling across these different amino acids. Okay. Unlike our carbohydrates, we actually don't have to get down to individual monomers to absorb proteins. So we actually are able to pull across some dipeptides and some tripeptides. So we can pull little groups of 2 or groups of 3 amino acids using these different membrane proteins as well. So if we do that through this process of active transport using energy to pull things in, we would then continue to break down those dipeptides and tripeptides to amino acids within the cell. So once we absorb a dipeptide or a tripeptide into one of these intestinal cells, we'd have enzymes inside that cell that can break it down further before we send it to the blood in its monomer amino acid form. Yeah. Cool. So next, we're going to think about our lipids. They're going to work a little bit differently. So it's going to be worth our time to think about how lipids work differently than our carbohydrates and our proteins, because this is going to be important for the digestion process. So go ahead and look at these different potential kind of chemical characteristics, physical properties that we might have. Think about which of these is true as a kind of definition of how lipids writ large, so like lipids in general, work. Okay, cool. Yeah. So in general, the way we group together our lipids, whether they're fats or oils or waxes or steroids, is that in general, lipids are non-polar, right? So they're mostly made of carbon and hydrogen, which have equivalent electronegativity. So we end up with all these non-polar bonds in our generic lipid. Right, I'm not going to draw a big old lipid, but we see something like this if we're looking at a lipid. And this affects how they operate in water, right? So our carbohydrates have a lot of like little alcohol groups on them or hydroxyl groups, right? So they mix well with water. So carbohydrates are hydrophilic. Proteins also can have polar groups, right? They have a lot going on depending on what amino acids are involved, but they're going to mix okay in water as well, depending on the specific protein. But lipids in general aren't. So our lipids, because of these non-polar bonds, are usually hydrophobic. So we do have some lipids that are amphipathic. So you weren't wrong if you were associating that word with lipids, but those are specific types. Specifically, usually when we're thinking about lipids that are amphipathic in biology, we're usually thinking about our phospholipids that form our cell membranes. But if we're thinking about lipids that you might be eating, like fats, we're going to be thinking about this nonpolar hydrophobic type. So this means that when you eat a fat, it's not mixing well with this watery environment in your body, right? There's a lot of water in all these fluids in the stomach and that we're secreting into your small intestine, your large intestine, et cetera. So this is going to create kind of a problem for us for digestion. We're going to solve it. But we have to operate a little differently than when we're thinking about our carbohydrates or proteins that have different chemical and physical properties. Okay, so if we're thinking about a typical diet, you're actually going to be eating about 50 grams per day of lipids. Obviously, that varies depending what your favorite foods are, depending whether you're tracking this stuff and trying to adjust it. But regardless, when we're thinking about the lipids you're eating, they're mostly going to be triglycerides, like about 90% of them. So that's what we're going to focus on as our example as we think about lipid digestion. So a reminder what a triglyceride is like, right? So we have our glycerol backbone, and then we have 3 fatty acid chains coming off of it, right? So it looks kind of like a flag. Okay. So that's our fats and our oils usually. So we've got this problem because those fatty acid chains here are just made up of carbon, hydrogen over and over and over again, nonpolar, don't mix well in water, not water soluble. Okay. So they're not going to mix in to our chyme the same way. They're going to form like fat globs, right? Same thing's going to be happening as they move into the intestine. So we've got these basically droplets where we have all of those little lipids grouping together in a big ball, right? Which means that if we're trying to break down a lipid or a triglyceride, we're not going to be able to get to those ones in the center because we're only going to be able to use our enzymes on stuff we can touch, right? So we can only get to the stuff on the outside of this fat droplet, all right? So this is the first problem that we're going to try to be solving as we're thinking about lipid digestion. Okay. So a little bit about those enzymes that ultimately will attack the individual triglyceride molecules. They're in general going to be referred to as lipases. The ones that we're going to be interested in are generally secreted from the pancreas. A little bit of lipid digestion does happen earlier on in the process, but not super substantial. Okay. Which means that when we think about lipid digestion, we're usually going to be focusing on the small intestine, starting with the duodenum, where we have those pancreatic juices coming in. Okay. So these lipases, these enzymes, right? So that ascending that break down lipids, telling us what they do, right? They can only act on triglycerides that they can touch. So they're only going to be able to attack molecules that are at the edge of a fat droplet. So what we're going to do in order to make our digestion of lipids more efficient is we're going to break up those fat droplets, right? So we have to take our larger fat droplet and break it down into smaller ones so that we have more surface area, so that all of these triglycerides, all of these lipids are exposed to the enzymes that we're using to chemically digest them. So that process of breaking down a big blob of fats, a big blob of lipids, is called emulsification. Right, so we're going to be emulsifying our lipids. So we're going to be making them more mixable into this watery environment in our body through the use of bile salts that are made by the liver. Okay. So here's what we see happening when we focus in on those bile salts. Okay, so they're coming from our liver, one of our accessory glands. So we know that the liver is made up of a bunch of different cells, including hepatocytes, making our bile. That includes our bile salts. We send those bile salts in bile through our biliary system. So we're thinking through the various branches of the bile ducts into the duodenum, coming in at that same place where we have the pancreatic duct coming in. Okay, so bile salt is made from cholesterol, right? So here we see that typical kind of steroid form. It looks like we've got kind of like 3 or 4 rings jammed together. And we got an extra little piece here that's going to help our bile salts act the way they do. Okay. So our bile salts are going to be amphipathic. So our bile salts are going to have this property where they have a hydrophobic side, right? So a part that's nonpolar and a part that is hydrophilic, right? So a part that is polar and is going to mix well with water. So when we say that we're going to use these bile salts to emulsify fats, the way we're going to be emulsifying them, the way we're going to make them more mixable in water, is we're going to be taking that hydrophilic side, taking that hydrophobic side. It can touch lipids because they're also hydrophobic, and that like attracts light. But on the opposite side, where we have that hydrophilic side, that part can touch water, right? Water is polar. Now we have these polar hydrophilic groups on the outside of our bile salt. So that's going to mix fine into this watery solution. So that's how we're going to be able to make our fat droplet, or this large fat globule, a little smaller. So we can see that happening here in this diagram. So we have our big fat globule where we have this problem where we would only be able to attack the edges with our lipases, right? So we're not going to be able to effectively get to the center of this fat globule. So we wouldn't be able to really digest and then absorb those fats inside. But when we put in our bile salts coming from our liver, here we can see how that big fat globule is now slightly smaller fat droplets. They have more surface area. You can see how that bile salt is bound to our fat droplet. So here we see those little blue dots are the polar side. The yellow side is that part that is able to touch our non-polar, usually triglycerides, meaning that we now have enough surface area that our pancreatic lipases are going to be able to attack substantially more of these triglycerides that we've eaten. Okay. So once we're thinking about that chemical digestion process actually breaking down our triglyceride, that's the job of the pancreatic lipase. So we turned our fat globule into smaller fat droplets using bile salts from the liver. Now we're going to actually break up our triglyceride. So it's going to break into its separate pieces. So basically, we're trying to take this triglyceride and peel off those fatty acid chains, right? So we're going to see our pancreatic lipases chopping fatty acid tails off. Okay. So some of these fatty acids and monoglycerides, so we are also going to be working with that glycerol backbone, right, are going to be absorbed into the cell, so across the wall of our small intestine. Others are going to stick around for a while in the form of micelles. Okay, so micelle, we mentioned like once in probably like one slide in A&P 1. Maybe you've talked about it in some of your chemistry classes, not sure. But what a micelle is you know how our phospholipids arrange into that phospholipid bilayer that forms our cell? A micelle is similar, but instead of having 2 layers, right, which gives us the ability to have a center with a watery environment, our micelles basically have just all that nonpolar stuff filling the center as nonpolar things attract to each other. Okay, so some of our fatty acids and monoglycerides are immediately getting absorbed into our cells. Some are sticking around in the form of these micelles, where we have these triglycerides that have been partially digested in terms of they've been broken up a bit, but we haven't absorbed them yet. Okay. So there's going to be an equilibrium happening in your system here, where these micelles are going to be sticking around as you absorb some of these fatty acids and monoglycerides. So that's what we see kind of happening here. Okay. So here is what we're just looking at, our fat droplet, right? So we can see those bile salts still on the outside. And we can see that we have some triglycerides here. Just for kicks, we put some other types of lipids like our phospholipids in this fat droplet. Our lipase is coming in, starting to break stuff up. All right, so now we see individual fatty acids that we've peeled off. And the monoglyceride technically still has like a fatty acid chain still on there with that glycerol backbone still attached. So that's why it has this kind of T shape. Okay. So some of those are getting absorbed into the absorptive cells of our small intestine, just kind of directly going across. Right? We don't need any transporters. We don't need any proteins necessarily for this part because they're nonpolar. Our cell membrane, by and large, is nonpolar, so they can diffuse across. Right? We don't need to use any ATP. We don't need to use any energy here for this process. This is just simple diffusion. And then some of these broken down pieces are still hanging out here in the lumen in the form of micelles. As we move forward and absorb more and more, those micelles will get smaller and more of these fatty acids and monoglycerides will get absorbed. Okay. So we're not done with our lipids when we pull these fatty acids and monoglycerides into our intestinal cells. Okay. So once we pull them into the cell, we're going to basically reform a bunch of our larger lipid molecules. So we broke them down, pulled down the broken down pieces into the cell, and now we're going to use our endoplasmic reticulum, specifically our smooth endoplasmic reticulum, right, one of our organelles, to reform them into triglycerides and some other types of lipids. From the smooth endoplasmic reticulum, they're going to get passed to another organelle, the Golgi apparatus, and they're going to get packaged into this form called a chylomicron. So we're going to have these chylomicrons as kind of our end result here in the absorptive cell. We're still not done. Now we're going to take that chylomicron, we're going to secrete it through exocytosis across the opposite side of the absorptive cell. So now it's in the interstitial fluid, so kind of near the blood, but it's not going to go into the blood. Instead, it's going to go into the lymphatic system. So that's why I pointed out that green lacteal on the inside of our villi when we were looking at the wall of the small intestine, because that's where our lipid absorption is happening. Okay, so here we see that process, right? So we pulled in fatty acids, we pulled in these kind of T-shaped monoglycerides. And this can just happen by diffusion, get inside the cell. Here we see that endoplasmic reticulum, specifically the smooth part, so the one that doesn't have ribosomes. And we can see the Golgi apparatus here as well. So they're acting together to take those individual fatty acids and monoglycerides, make larger triglycerides again, and put them into these chylomicrons. Okay. So that big blue ball is our chylomicron. We can see it's surrounded, right, by some membrane here, right? So it's in a bag from the Golgi apparatus as it packaged and processed this stuff. There would be some protein also in this chylomicron, making up part of it. They're not the main way we absorb proteins, but they are involved in this process. Then we take this chylomicron, move it to our basolateral membrane, where we can see that bubble bursting. So this is our process of exocytosis, and the chylomicron is now in that interstitial fluid. Okay. So once it's in the interstitial fluid, it bypasses the blood, And instead of going into the blood, it goes into our lacteal, which is part of our lymphatic system. So we have lymph, its own kind of separate fluid, running parallel to our arteries and veins, going through these lymphatic veins that ultimately do jump back into your cardiovascular system, kind of right before you get to the heart. So they're going to dump into some of the veins going into the heart as you enter the thorax. So we've had a real kind of roundabout way of digesting and absorbing our lipids, largely because of this difference in their chemical and physical properties. It means we can't do what we would usually do if we had something that mixes well into the watery environment in the lumen of the GI tract, and also the watery environment inside our cells and in our blood, right? So it's actually dangerous if you have a bunch of lipids building up in the blood, right? That's how we get like cholesterol buildup, kind of closing over the walls of our arteries and stuff. So we don't want lipids building up here in our blood. So it's good they're going into our lymphatic system. So here we can see kind of our circulation of the bile salts as they're doing this process, and then also getting reused, right? So you don't need those bile salts once you've broken up the larger fat droplet. So we're not going to excrete them all out. We're going to try to take some of them back up. So here we can see the liver making those bile salts, sending them down into our biliary system. Ultimately, we're focusing on the common bile duct as it enters the duodenum. Here they are in the duodenum, hanging out, waiting for some lipids to emulsify. They emulsify them. The constituent parts of those lipids get absorbed into the small intestine. Doesn't take super long to do that. Your small intestine is pretty long relative to how much space you should need to do that absorptive process. So those bile salts are passing from the duodenum to the jejunum. And by the time we get to the end of our small intestine, the ileum, hopefully we don't need them anymore. So we are going to reabsorb at least some of them so that we can reuse them. So this is where it comes in handy that the veins attaching to our small intestine and also to parts of our colon are part of this hepatic portal system, right? So veins in the abdomen, for the most part, don't go directly to veins that go to your heart. First, we're funneling them all through the liver, right? So we see them going through those veins into the hepatic portal vein, into the liver, where we can reuse some of these bile salts. So we can make new ones, but we want to be efficient with this process. All right. So the main things that you're going to be eating and working on absorbing into your body are those carbohydrates, proteins, and lipids. But we're also going to need some vitamins, some minerals, and water. Okay. So when we think about these other kind of like micronutrients that we need, we're going to think about their physical and chemical properties as well. So when we think about vitamins, we tend to divide them into fat-soluble vitamins, so ones that have more of those like hydrophobic properties and will mix in with lipids, versus water-soluble vitamins that are going to mix in with that watery environment in the lumen of our intestines. Okay. So when we think about fat-soluble vitamins, the main ones we're thinking about are vitamins A, D, E, and K, and they get absorbed basically along with the lipids. So that process that we just looked at, where we break down our larger fat globule into smaller lipid droplets, lipases attacking our triglycerides. We're not going to have to break up a vitamin, so it's just going to hang out while the lipases are doing that. But they're going to get incorporated into the mice cells that are swimming through our small intestine. They're going to get absorbed into our intestinal cells, and they're going to get processed inside those intestinal cells into those chylomicrons that ultimately go into the lacteals and the lymphatic system. Okay, so they follow along with our lipid process. Our water-soluble vitamins don't follow our lipid process because they're water-soluble, hydrophilic. They're going to have transport proteins associated with them. So they're going to have proteins embedded in our cell membranes that allow us to pull water-soluble vitamins into your body, into those cells. One particular one that we've mentioned before is vitamin B12. So vitamin B12 is important for like cell proliferation. We mentioned that missing vitamin B is one of the types of anemia we can have, our pernicious anemia, because we can't make enough hemoglobin. We also mentioned vitamin B12 as we were thinking about this stomach, because one of the other things that's getting secreted into our gastric pits is intrinsic factor. So intrinsic factor, this guy here, that's coming from the gastric pits. So in addition to hydrochloric acid and pepsinogen, I got intrinsic factor going into the chyme, which is great. Chyme ends up in the small intestine, which is where most of our absorption is happening. We need this intrinsic factor to be bound to vitamin B12 in order for us to absorb it. That's just kind of the way the transport proteins for that specific vitamin work. Other water-soluble vitamins would have their own transport proteins that allow their absorption, but don't tend to need another extra factor to work with them. When we think about minerals or electrolytes, we're usually thinking about like sodium, chloride, potassium, are friendly ions that we've seen throughout this course. So sodium is usually going to get dragged along with water. We know that often happens as we have the action of sodium-potassium pumps. We're going to see active absorption of sodium as well in the jejunum, the ileum, and then also in the colon, right? So our carbohydrates, our proteins, and our lipids are mostly being absorbed in the small intestine. But when we think about sodium and we think about water, this can happen further on. Chloride is a negative ion. An anion goes with the sodium. Potassium absorption is kind of passive, but how that exactly works is going to depend on exactly what you've been eating and exactly what the concentration of potassium is in your foodstuff and in the lumen of the GI tract, because we can also secrete potassium. So when we have low potassium environment in the lumen, we may end up secreting rather than absorbing potassium. Okay, so we'll stop there for now. And we've got like 2 slides, a couple more slides on this before we move and think about our next packet on Monday.